Hello students, now we will discuss about coelom, which is also called as body cavity. So coelom also called as body cavity. Coelom is the space between body wall and also alimentary canal or gut wall. Coelom is the space between body wall and the gut wall, which is also called as alimentary canal. Here there is a rule that coelom has to be lined by mesoderm. Then only we will call that space cavity as coelom. So based on presence or absence of coelom, there are different types of categories of organisms. Based on presence or absence of coelom, in animal kingdom, organisms are termed as, first one, acoelomates, in which the coelom is absent. Example for such types of organisms are Porifera, Coelenterata, and Ctenophora and Platyhelminthes. First one, acoelomates. The organisms in which coelom is absent. Example, Porifera, Coelenterata, Ctenophora, and Platyhelminthes. So during embryonic development, we know that after fertilization of the gametes, zygote is formed. Zygote is divided into many number of cells. Then we give different terms as 8 cell stage, 16 cell stage, etc. So during those stages, when the total mass of cells has reached 16 to 32 number of cells, then we give a term as morula of the embryonic development. After morula, the next stage is blastula. The next stage is gastrula. During the gastrulation time, this coelom is formed. We'll write that point. During embryonic development, the stages are, first one, morula, second one, blastula, third one, gastrula, fourth one, organogenesis, means here the body organs are synthesized. Before this, we see gametes which are fused, means female gamete plus male gamete. When they are fused, they lead to formation of zygote. Zygote leads to formation of 8 cell, next 16 cell, next 32 cell, etc. By the time here of morula, we see 16 to 32 number of cells. In case of zygote, we see only 1 cell. In case of blastula, the total number of cells will be increased by 100 number. It can be 100 or more than 100. 100 number of cells. So in such a way, the stages will be proceeded. At this gastrulation stage, coelom is formed. But the coelom has to be lined by mesoderm. Then only we give a term as coelom or body cavity. Coming to diagrammatic representation, if we do the transverse section of the gastrula, then outermost layer is nothing but having ectoderm. Next, In the inner gut wall or alimentary canal, it is lined by endoderm. If the middle layer is not having any mesodermal cells or layers, then it is called as acelomate. Then what is present here in the gap? Is that gap is present like that only? Here will be present mesoglia, a fluid which is resembling that of mesoderm. Some fluid like substance will be present. That is termed as mesoglia. This outermost one is ectoderm. Inner one is endoderm. So in embryonic development, we see three different germinal layers which are called as ectoderm, mesoderm and endoderm. In case of acoelomates, mesoderm is absent, but the cavity is filled by mesoglia, some fluid-like substance. Then what is meant by, what are the uses of coelom? Coelom uses. So it provides a space for the fitting of the vital organs. What are vital organs? Vital organs are those that does the metabolic activities, just like in case of human body, liver, digestive tract, kidneys, pancreas, etc., heart, all these structures are considered as vital organs. The structures that does metabolic activities. Next, coelom provides flexibility to the body. Next, when the coelom is filled by coelomic fluid, that type of organisms are also present. This flexibility, majorly it is seen in case of, due to coelomic fluid in case of ascaris or nematodes or Nematihelminthes. So in ascaris, coelom is filled by coelomic fluid and gives flexibility to body. In some types of organisms, complete coelom or body cavity is not lined by mesoderm, but the body cavity or the coelom is having a little bit patches of mesoderm cells or tissues. Such types of organisms which are not having the coelom completely lined by mesoderm are termed as pseudocoelomates. Example is Nematihelminthes or nematodes or Aspelminthes. Next is pseudocoelomates. The organisms in which coelom is not completely lined by mesoderm but have mesoderm tissue patches. If you see the diagrammatic representation, if this is the gastrula stage, during gastrulation, we said that during that embryonic development, coelom is formed. So this will be the ectoderm, and this will be the endoderm. Now, in the middle area, here and there will be mesodermal cells or patches. So if the mesodermal distribution in the coelom is like this, then we call such organisms are pseudocoelomates. Example, Nematihelminthes. Next, if the organism is having well-developed coelom, means complete coelom is lined by the mesodermal layer, then such organisms are termed as coelomates. The organisms in which the coelom is completely lined by mesoderm. So basing on the embryonic development, how this coelom is formed, it is of two types. On the basis of coelom formation in embryonic development, the coelomates are of two different types. First one, schizocoelomates. Second one, enterocoelomates. In both cases, the coelom is present. But how the coelom is formed in case of schizocoelomates and how the coelom is formed in case of enterocoelomates. Under schizocoelomates, we will write examples like Annelida, Arthropoda, and molluscans or mollusca. Now, in case of schizocoelomates, if this is the embryo, this is the ectoderm, and this will be the endoderm. Now, any type of embryo or the gastrula form will be having one opening. That opening is called as blastopore. For suppose think this is the structure. Its arrangement will be like this. That opening will be converted into mouth in some types of organisms and into anus in some types of organisms. So, if the opening or this structure is called as blastopore. Now here we have endoderm and also one blastopore. So we'll label this one endoderm, this one as ectoderm. So at the junction of this endoderm and this blastopore, here arises a mass of mesodermal cells. So these are mesodermal mass or cells or tissues. Now, these will be responsible for the formation of coelom in the later stages. For suppose, this will start forming a central coelom-like structure and it will be increased in size like this. So, in the later stage, it will be formed into here forms the mesoderm. This will be the coelom. So this one and this one are considered as coelom. Now this lining, this one is termed as mesodermal lining. So right here, this is mesoderm. In case of schizocoelomates, if we want to frame out a sentence, how do we frame? In the organisms in which mesoderm is splitted into means cavity-like structure and the coelom is lined by mesoderm. So schizocoelomates will write a definition here. The organisms in which coelom is formed by splitting of mesoderm. You write mesodermal mass. Now, this happens in case of protostomes. There are some types of other organisms. Those are nothing but deuterostomes. In case of protostomes, the blastopore is converted into mouth-like structure. But in case of deuterostomes, the blastopore is converted into anus-like structure. Next is enterocoelomates. In some types of organisms like echinoderms, hemichordata and chordata, during gastrulation stage, some mesodermal cells will be accumulated on the primitive gut wall or the early developing gut or alimentary canal. Then later this mesoderm will be pinched off along with the archenteron cells or the primitive gut wall and it forms into a coelom. Now we will see that one in detail. The organisms in which the primitive gut wall or archenteron is pinched off along with mesodermal cells when endodermal cells are protruded towards ectoderm. Now the structure looks like this will be the ectoderm, this will be the gut lined by endoderm, this is connected to the blastopore structure. Now some of the endodermal cells will be protruded outside like this. Now these cells are connected with the originated mesodermal cells. Later they will be pinched off and this will be converted later into the same type of structure like ectoderm, and here will be the gut, endoderm, again the formation of coelom. So this will be the coelom. So in case of enterocoelomates, the archenteron or the alimentary canal gut is involved in formation of coelom. In case of schizocoelomates, the mesodermal mass, it will be converted into coelom. So examples for enterocoelomates are echinoderms, hemichordata and chordates. This is about coelom.